In 2015, researchers studying octopus brains noticed something very strange. The genetic instructions inside their nerve cells didn't match their DNA. At first, it looked like an error, maybe a glitch in the data, but it wasn't. It turned out octopuses and their relatives were actively rewriting how their own genes were being read. And not across generations or through slow evolutionary change. They were doing this while they were still alive. This was the first time scientists had ever seen an animal do something like this at such an extreme scale. And if that's true, it suggests that cephalopods may represent a fundamentally different approach to evolution than anything else we've seen in the animal kingdom. So what does it actually mean to change how your genes are read? Why would an animal want to do this? And if some creatures really can adjust themselves within a single lifetime, what does that say about how evolution actually works? For most animals, the rules are simple. Your genetic code shapes how your body grows, how your brain is built, and even how it responds to the world around it. That code is inherited at birth, and while you can learn and adapt to what you do, the genetic instructions your brain is built from aren't supposed to change. And if they do, it happens slowly through generations of evolution. That's the framework that biology has worked with for millions of years. And it explains why we're often left with things like phobias, traces of our past that once helped our ancestors survive, even if they don't make much sense in the modern world. And for almost every animal we know, those rules hold. Because if an animal could change those instructions when it wanted, if it could tune its nervous system to the world it lives in, not over generations, but in its own lifetime, that wouldn't just be unusual. It would break one of the deepest assumptions we have about evolution itself. And yet, in cephalopods, research showed that might be exactly what was happening. To understand what they are doing, we need to zoom in, down to the level of the genetic instructions themselves. Typically, DNA acts as a blueprint. It contains the instructions for how your body is built and how it functions. To use those instructions, your cells make a working copy, a molecule called messenger RNA, or mRNA. That mRNA is then used to build proteins, which do most of the actual work in the body. In most animals, that RNA copy is supposed to be faithful, but in cephalopods, that isn't what happens. When researchers started looking closely at cephalopod neurons, they didn't just find a few small deviations. They found widespread changes across their entire nervous system, happening after the RNA was already made. What researchers were seeing had a name, and it's called RNA editing. And in most animals, it's rare, very tightly controlled, and limited to a handful of sites where small tweaks help to stabilize critical proteins, especially in the brain. But in cephalopods, something very different was happening. See, we edit RNA at a few dozen, maybe 100 sites. For mice, it's similar. For flies, maybe close to about 1,000. But in cephalopods, around 80,000, with most of the edits concentrated inside their nervous system. And almost all of these edits followed the same pattern, a one-letter change, adenine to inosine, which is instead read by your cells as guanosine due to their similarity in structure. Not a rewrite of the entire code, but a precise tweak to how parts of it are read. It's subtle, but enough to change how neurons behave. In one set of experiments, researchers raised two groups of octopuses in different water temperatures and recorded how their genes were being edited. Then they did something simple. They switched the temperatures. And as the environments changed, so did the edits. In colder water, certain neural proteins were edited more frequently, and it seems their nervous system was being tuned to the new environment in real time. And how long did all of this take? The answer is 4 days. 4 days to make sense of their new environment and modify themselves biologically to be better suited for it. Understandably, they were very excited and began testing what these edits actually do. In some cases, they found the edits made neural pathways faster by fractions of a millisecond. In others, it altered how easily neurons release signals, which changed how readily a response could be triggered at all. Tiny differences on paper, but potentially huge consequences for how these animals respond to danger and environmental pressures. And here's the other thing. Unlike DNA, RNA is much more flexible. You can make changes, and if they stop being beneficial, they'll naturally fade away. Which matters in a world where conditions don't stay the same, with things like seasonal or rapid temperature changes. So, if RNA editing is this powerful, if it allows an animal to tune its nervous system without committing to a single biological solution, then the obvious question is, why don't more animals do this? Why hasn't this strategy spread across the animal kingdom? The answer to that seems to be that this flexibility comes at a steep price, because rewriting RNA at this scale doesn't just change how neurons behave, it changes how evolution itself can work. As far as we know, this ability only appears in soft-body cephalopods like octopuses, squid, and cuttlefish, who split off hundreds of millions of years ago from their relatives like the nautilus and Aplysia, and instead went down a very different path. For simplicity, when I say cephalopods from here on, I'm talking about this soft-body group. Whatever happened along that branch in history fundamentally changed how they use their DNA. And in doing so, they sacrificed something critical: genetic variation. That may sound counterintuitive at first, but to keep RNA editing possible at this scale, the underlying genetic sequence has to remain almost perfectly stable, because the thing that allows them to edit RNA relies on precise molecular structures. And if the DNA around those sites mutates too much, the editing breaks. Think of it like a finely tuned watch. You can adjust how fast it runs and fine-tune its timing, but only because the inner mechanism, the gears that make those adjustments possible, stay exactly the same. If those inner parts were constantly changing, the ability to adjust the watch would disappear. And that is what cephalopods have locked down. They preserve the parts of their DNA that makes RNA editing possible. So cephalopods didn't give up on traditional evolution, but rather they constrained it selectively, trading long-term genetic flexibility in certain genes for short-term reversible control over how those genes are expressed. So this begs the question, what kind of world makes this strategy worth it? The kind of world that changes faster than evolution can keep up. See, for most cephalopods, life begins with no guarantees at all. They hatch in the thousands and, depending on the species, even millions, and almost immediately they're carried away by ocean currents. Some drift into warmer waters, while others into colder, darker environments, different pressures, different predators, and there's no way to know in advance which world an individual will end up. For many species, the vast majority don't survive this stage, and the ones that do aren't necessarily the strongest. They're the ones that can adjust most efficiently to wherever they land. And even for those that survive, time is short. Many cephalopods only live for a year or two, often less, during which they need to hunt, learn, and reproduce. There's no long childhood to safely test the waters because they don't rely on large social groups, and they don't grow up with parents to learn from. Each individual has to solve its own problems alone. In that kind of life, it can be almost impossible to find one or few consistently successful traits to develop over generations. Instead, what matters is being able to tune yourself within a single lifetime to the world that you actually end up in, and RNA editing gives them that option. It's an almost alien concept when we compare it to our own, a different evolutionary path and a different approach at adaptation itself. So what can we learn from this? Not just that cephalopods are strange, but something quieter and more powerful. Cephalopods show us that there's more than one way to adapt. Much of what we understand about this today comes from the work of neuroscientist Dr. Rosenthal and his team, who have spent decades studying RNA editing in soft-bodied cephalopods. And Dr. Rosenthal has pointed out that understanding how these animals edit RNA in nerve cells could one day help us design therapies that work the same way. Not by permanently rewriting DNA, but by temporarily adjusting how cells behave. For example, by dampening inappropriate pain signals in chronic pain, or choosing how neurons communicate without locking those changes in forever. In that sense, RNA editing would be very different from tools like CRISPR, which makes permanent changes to DNA. As Dr. Rosenthal puts it, if you make a mistake in RNA, it's not nearly as dangerous, but if you make a mistake in DNA, you're stuck with it. None of this means we're anywhere close to using RNA editing like cephalopods do, but it does suggest something important. For hundreds of years, we assumed there was only one way to adapt: to rely on mutation to change the genetic code, to wait, and to let natural selection do the rest. Cephalopods remind us that this wasn't the only option. They didn't evolve to become perfect, they evolved to stay adaptable. It was a deal evolution offered the cephalopods millions of years ago, and one they decided was worth keeping. And in a world that refuses to stay still, that might be one of the strangest and most powerful strategies evolution has ever offered. If you enjoyed this video and want to see more like this, please feel free to like and subscribe, and I'll see you in the next one.